Okay then, so hello everyone and thanks for joining the 12th Nuvisan Talk. Today we will talk about drug discovery for venture capitals, how to de-risk your investment. So we know VCs are assuming most of the risk today in drug discovery, and with the high attrition rates that we observe in this market, it's key to find ways of maximizing your chances of success. So finding a partner that offers the highest scientific quality possible and that is willing to have a skin in the game is a starting point. So next slide, please. So the speakers for today are Charlotte Kopitz. She's head of therapeutic research, and she'll talk about de-risking the project scientifically. Charlotte is a biologist by training. She did a PhD and postdoc in experimental oncology. In 2008, she took over the lead of the in vivo laboratory in oncology research at Bayer. In 2019, she took over the lead for pharmacology of the Innovation Campus Berlin at Bayer. And in 2020, when Nuvisan acquired ICB, she joined as EVP and head of therapeutic research. And the second speaker is Arnoud Huisman, managing director and chief strategy officer, and he will discuss collaboration models between CROs and venture capital and different risk sharing options. And a bit about Arnoud, he has over 25 years of experience in leadership positions in life sciences, including pharma, CROs, startup, and venture capital. And he joined Nuvisan in August 2020, just right after the acquisition of Innovation Campus Berlin. So next slide, please. This is the agenda for today. We will start with the process from target understanding to clinical candidates. We'll discuss asset indication mapping, the design of the optimal compound, then how translational pharmacology research is key for sound clinical development. We'll discuss biomarkers, discovery and validation, and we will discuss how to increase the market size with finding the right drug combination partner. And then lastly, we will talk about different combination models between CROs and venture capital, and we will finalize the session with a Q&A. Next slide, please. Some house rules. All of your microphones are muted by default, so please use the Q&A box below here, not the chat, the Q&A box for your questions. And please, I want this to be really a bidirectional session, so feel free to ask really all your questions. There are no stupid questions, and I really want that your time spent here is really time well spent. And the last thing is that this session will be recorded. After the event, you will receive an email with a link that you can share with your colleagues so they can see if they could enjoy today. Next slide. So now I would like to start with a question to all of you. So we would like to know who you are. Are you a venture capitalist, company builder, scientist, students, other? We will leave you a minute or so to reply, so everyone can get a better feeling of who's the audience. And I would like this moment to really align with everyone and highlight that this webinar is mainly focusing on venture capitalists and company builders. So let's see, replies are coming in. Please, yeah, now let's see the replies. Okay, so we see here that 30% are both venture capitalists and company builders. Great. Many are scientists, some students, and then others. Well, thank you, everyone. Next question for you. Next slide, please. Have you worked with Nuvisan before? So here there's a display of many different answers. So some are yes, some are no. Some are like if you work with Charlotte, Arnoud, or myself before. So feel free to choose the answer that better suits. We'll wait for a minute to get all replies in. So this is for everyone to know really who the audience is and really better understand. Okay, so now the answers. So this is good. So most of you never worked with us before. So I really think it's the place for you to be. So great. Let's close and continue. Next slide, please. So now I'll hand it over to Arnoud.
Yeah, so thank you very much, Carmen. And it's good to see that at least 3 people have not worked with Nuvisan before, but know either Carmen, Charlotte, or me. So that's good. For all the others, thank you for joining. Good morning, I think, to our America friends, because there's quite some people from the Americas joining today. Good afternoon, of course, for everybody in Europe, and good evening for our Asian friends that are also joining. So hopefully we will have an interesting program for all of you all over the world. And we thought it might be a good thing to start with a little bit of the history of Nuvisan and how we have been building up the company over the last 43 years and make it ready to be also a good partner for venture capital. So like I said, 43 years ago, started originally as a lab, by analytical and through acquisitions, especially over the last 10, 15 years of units that used to be from Big Pharma, right? Like in 2011, the acquisition of a pharmaceutical analysis unit in Western Germany from MSD, also Galderma in the south part of France, a GMP unit, a proof of concept unit for respiratory patients close to Munich in 2019, and I think one of the bigger acquisitions in 2020 with the integration of the Innovation Campus Berlin, an ex-discovery unit from Bayer, where both Charlotte and I actually work, and we are presenting this today for you from this specific site. So this is the acquisition strategy that Nuvisan had over the last couple of decades. And of note, I think, is that we had a major partner joining us last year with ALS, giving us some more breadth to continue to grow to a full service partner for biopharmaceuticals, but also for venture capitals. So if you go to the next one, you will see that we actually offer the entire value chain, all the way from very basic research, target identification, all through the steps in hit identification, finding the leads, optimizing it, go to IND enabling studies, do the non-clinical, preclinical, tox, etc., all the way into first in humans and proof of concept. And along these later steps, also provide the pharmaceutical services and the analytical services. So this is really all within Nuvisan command, within Nuvisan management. So it's really one team, one partner. And by that, we can work with you to go from the discovery all the way to the patient, right? And take all the important step in the value chain, helping you to develop your asset. We can, during this development, optimize the drug development with you, and especially for venture capital, of course, very important, de-risk the projects. Of course, along the way, we will hit surprises, right? This is discovery. That will happen for sure. And because we have here in Berlin, at least from an innovation perspective for the discovery side, we are all in one building, right? All the first steps to preclinical IND enabling studies, we are all in one building, so that makes it relatively easy to cope with the changes that we always will find during the project. So I think this is an important element for you to realize. And given the fact that we are all ex-Big Pharma scientists, right, that sort of makes us kind of a startup, because we're only here for 2 years yet. But at the same time, it's a startup with 500 people that have done this work for years and years and years, and know what it takes to maximize the success for a program that will lead to a new molecule. And a very important element of that is the compound library, where we have access to 3 million well-characterized chemicals that we have in there, next to the life sciences database, right? So we can really do a smart step into the drug development. Now, if we try to put that into one graph, next please, Charlotte, and how we do all this drug discovery under one roof. You see the flow here from lead discovery into high throughput screening, do the prioritization, do the design and the synthesis in our medchem department, together with Charlotte in the therapeutic research department, to do the in vivo, in vitro biomarkers, the PK studies, the DDI, the safety, etc., etc. So this entire flow, one team, one partner, one solution, and we can help you to de-risk your compound. Now, it would be very dangerous for me if I'm actually going to explain how that works. Luckily, we have Charlotte, and she will tell you exactly how this is done. So please, Charlotte.
Thank you. So I would like to continue. And first, we, of course, try to think about how can we help to increase the value of your investment by the services that we can offer. And I would like to briefly start with the next 2 slides on one side of the coin, which is finding the right clinical molecule to go forward, because it's as obvious as better your clinical molecule is, the higher the chance of success in clinical trial will be. So what we can do, as Arnoud just mentioned, so we can start with the screening and hit finding to come up with a hit molecule, which is the first tool to prove your hypotheses. Of course, this is not the way how to go forward directly. So we need to design and select an improved molecule in order to have the best starting point for compound optimization, because this avoids a multi-parameter optimization, which means you save time and finally also costs on long run going forward towards clinical studies. And the good point is with our services, as just elucidated by Arnoud in the slide before, we have everything in place which you need in order to come from a hit molecule to a lead molecule and also to go forward to have the final clinical molecule, because this is an intense interplay between DMPK, pharmacology, and chemistry, and of course the colleagues who are performing the high throughput screenings in order to have a compound with a sound and means of potency, efficacy, selectivity, metabolic stability, but also solubility. I would like to explain you why it is important once you have a lead molecule, which so far already has the activity which you like, to further invest some time to come to a clinical molecule or an optimized compound. So the lead molecule could still harbor potential risk factors for further development of a compound, which means it can have some off-target activity which you need to get out of the molecules during optimization. It can have a weak DMPK profile, leading that you can't achieve efficacious levels. It could be that it has a low potency or poor efficacy, which result into the same, that you would most likely not be able to achieve the necessary levels of the compound in human patients. And of course, it could be that the on-target activity is not as expected. So in order to optimize the compound, it of course takes time and also some cost during preclinical development. But finally, you have then a lower side effect risk, which is of course beneficial. You have a higher likelihood that you really can achieve your biologically active dose in humans, which is then also reflected by patients' compliance. So if you come up with a less soluble compound, you sometimes have tablet sizes, which even humans in the first in phase 1 trials might not swallow. Then there is the cost of goods, which is directly connected to it. And finally, you have, if you have all those factors optimized, a higher chance of proof of concept or proof of mechanism in your clinical trials, which means for this time and preclinical cost, you really have a strong gain in value of your asset from the lead molecule towards to a mature clinical molecule. There are other presentations which focus more on the side of the molecule going forward. I would like now to come more to the biological side going forward, because out of the drug discovery campaign, you're coming out with a fit for purpose drug candidate. So it is exactly the way how you need it to go forward. And you usually also have an in-depth understanding of the compound's characteristics. On the other hand, in the clinic, you would like to have an evidence-based clinical study design to have a higher probability of clinical success. And translational research or biology research leading to understand the disease and disease hypothesis is helping to take this hurdle into the clinic. And this is exactly also something which increases the value of your asset and potentially also from your company. And this is done by understanding biomarkers to know the size of your indication space going into, to have the largest indication space you can identify.